Dear students, I welcome you all to the 10th session of Histology, Animal Behavior and Applied Zoology paper prescribed by the Mangalore University for 4th semester BSc Zoology. In this session, we shall consider the study of histological features of mammalian kidney. The mammalian kidneys are paired bean-shaped organs enveloped by a thin capsule of connective tissue. Each kidney is divided into an outer cortex and an inner medulla, and each kidney contains about 2 million nephrons. A nephron and the collecting tubule into which it drains from a uriniferous tubule. The renal hilum is a concavity on the medial border of the kidney. It houses arteries, veins, lymphatic vessels, nerves, and renal pelvis as well. The renal pelvis is a funnel-shaped expansion of the upper end of the ureter. So this is the renal pelvis, and this region is called renal hilum. And the outermost part is the cortex, followed by a medulla. It is continuous with the major renal calyces, which in turn have several small branches, the minor calyces. The renal medulla lies deep to the cortex but sends extensions, which are called medullary rays, into the cortex. The renal or medullary pyramids are the conical pyramids, as you can see here. These are the renal pyramids. Conical structures that compose of bulk of the renal medulla. Each kidney contains about 10 to 18 renal pyramids. Each pyramid consists primarily of the thin limbs of loop of Henle, blood vessels, and connective tubules. The renal papilla is located at the apex of each renal pyramid. It has perforated tip, the area caribrosa, that projects into the lumen of the minor calyx. The renal cortex is the superficial layer of the kidney beneath the capsule. It consists of primarily the renal corpuscles and convoluted tubules. Renal columns of Bertini are the extensions of cortical tissue between adjacent renal pyramids. Medullary rays are groups of straight tubules that extend from the base of each renal pyramid into the cortex. So this is the major calyx and this is the minor calyx. The renal lobe consists of a renal pyramid and it's closely associated with cortical tissue. The renal lobule consists of a central medullary ray and the closely associated cortical tissue on either side of it, extending as far as an interlobular artery. Its many nephrons drain into the collecting tubules of the medullary ray. The renal interstitium is the connective tissue compartment of the kidney. It consists primarily of fibroblasts and mononuclear cells, probably the macrophages. In medulla, it consists of two additional cell types. They are pericytes. These pericytes, as you can see here, they are located along the blood vessels that supply the blood to loop of Henle. The interstitial cells have long processes that extend toward capillaries and the tubules in medulla. The nephrons. Nephrons are the structural and functional part of the kidneys. The nephrons consist of renal corpuscle, the proximal convoluted tubule, and the distal convoluted tubule. The nephrons can be classified as cortical or juxtamedullary depending upon the location of the renal corpuscle. Juxtamedullary nephrons possess a longer loop of Henle than do the cortical nephrons and are responsible for establishing the interstitial concentration gradient in the medulla. The renal corpuscle consists of the glomerulus and the Bowman's capsule and is the structure in which the filtration of blood occurs. So this is the structure of the glomerulus and the Bowman's capsule. This complete structure is called the renal corpuscle. Let us now look into the structure of Bowman's capsule. The parietal layer of the Bowman's capsule is the simple squamous epithelium that lines the outer wall of the Bowman's capsule. As you can see here, the outer wall of the Bowman capsule is made up of simple squamous epithelium. The visceral layer, the glomerular epithelium, is the modified simple squamous epithelium composed of podocytes. Let us study about podocytes in detail in the coming slides. That lines the inner wall of the Bowman's capsule and envelops the glomerular capillaries. The Bowman's space, also known as the capsular space or urinary space, is the narrow chalice-shaped cavity between the visceral and parietal layers into which the ultrafiltrate passes. The vascular pole is the site on the Bowman's capsule where the afferent glomerular arteriole enters and the efferent glomerular arteriole leaves the glomerulus. So this is the afferent and this is the efferent. The urinary pole is the site on the Bowman's capsule where the capsular space becomes continuous with the lumen of the proximal convoluted tubule. The podocytes are highly modified epithelial cells that form the visceral layer of the Bowman's capsule. These podocytes have complex shapes and possess several primary processes that give rise to many secondary processes called pedicels. The pedicels embrace the glomerular capillaries and interdigitate, with pedicel arising from the other primary processes. Their surfaces facing the Bowman space are coated with podocalyxin, a protein that is thought to maintain their organization and shape. Filtration slits are elongated spaces between adjacent pedicels. Diaphragms composed of layers of filamentous material bridge each filament slit. The renal glomerulus is the tuft of capillaries that extend into the Bowman's capsule. You can see here this is the glomerulus. It's a tuft of capillaries that has entered into the Bowman's capsule. The glomerular endothelial cells, that means the cells of the glomerulus, form the inner layer of the capillary walls. And these glomerular endothelial cells have a thin cytoplasm that is thicker around the nucleus, where most of the organelles are located, and they also possess large fenestrations but lack the thin diaphragms that typically span the openings of the other fenestrated capillaries. So in the previous sessions we have also seen the meaning of fenestrated capillaries. That means the capillaries with holes, and the hole, the fenestrae here in the glomerular endothelium is about 60 to 90 nanometers in diameter. The basal lamina is between the podocytes and the glomerular endothelial cells and is manufactured by both cell populations. It is usually thick and contains three distinct zones. So this you can see here, these are the podocytes with the processes and a basal nucleus. This is present in the Bowman's capsule. Now this is the endothelium, that means the blood vessels or capillaries. Capillaries means they are the glomerular blood vessels. And in between you can see many fenestrations. Fenestration means the opening. So these endothelial cells are discontinuous. So there will be openings. So now in between these two layers, see the podocyte layer as well as the capillary, there is a small space. And in this space we can see three layers, three distinct layers. The lamina rara externa. So this is the lamina rara externa, an electron-lucent zone adjacent to the podocyte epithelium. So the layer which is adjacent to next to the podocyte layer, is called lamina rara externa. And lamina densa is the electron dense intermediate zone of amorphous material. Electron dense means there are a lot of electrons in this layer, so it looks a little bit dense. You can see here, it's the color is a bit concentrated here because of the accumulation of some amorphous substances out there. And the third layer here is the lamina rara interna. Interna means present inside. Is also electron-lucent. That means that it contains very less electrons adjacent to the capillary endothelium. So on the other side, both the sides you have lamina rara, but the position of this lamina rara and lamina rara externa and interna is classified based on the position of the layer. If it is present towards the outside, it is called externa. If it is present towards inside, then it is called interna. The mesangium is the interstitial tissue between glomerular capillaries. It is composed of mesangial cells and an amorphous extracellular matrix elaborated by these cells. So the space in between the glomerular capillaries are called as mesangium. The mesangial cells. The mesangial cells are phagocytose. They phagocytose large protein molecules and debris which may accumulate during the filtration or in certain disease status. They can also contract, thereby decreasing the surface area available for filtration. So this is the mesangial matrix. You can see here they are present in between the endothelium of the glomerular capillaries. And they also possess receptors for angiotensin II and atrial natriuretic peptides. The mesangial matrix helps support the glomerular capillaries. The structure of renal filtration barrier is composed of fenestrated endothelium of the glomerular capillaries, the basal lamina, that means the lamina rara and lamina densa, and the filtration slits with diaphragms between the pedicels. The function of renal filtration barrier is they permit the passage of water, ions, small molecules from the bloodstream into the capsular space, but prevents the passage of large and most negatively charged proteins, thus forming an ultrafiltrate of blood plasma in the Bowman's capsule. So the actual the function of filtration is monitored by these renal filtration barrier. The lamina rare contains heparin sulfate. So this layer contains a compound called heparin sulfate, a polyanionic glycosamine glycon that assists in restricting the passage of negatively charged proteins into the Bowman's capsule. They will not allow the entry of the negatively charged particles into the Bowman's space here. Okay. The lamina densa contains type 4 collagen, so this layer, which acts as a selective macromolecular filter, preventing the passage of large protein molecules. The protein molecules with molecular weight more than 69,000 daltons into the Bowman space. Actually, those proteins and other substances will be present here in the capillary lumen, and it has to pass through this fenestrated endothelium and pass through this lamina rara and densa, then reach the Bowman space. So here the Bowman space acts as a major filter that will not allow the negatively charged proteins as well as the proteins having larger molecular weights. The next structure that we are supposed to focus here is the proximal convoluted tubule. So after the glomerular apparatus, we are supposed to study about the structure of proximal convoluted tubules. The proximal convoluted tubule, so you can see here, this is the proximal convoluted tubule. Convolutions, the special kind of arrangements like this you can see here. And proximal means it is the first convoluted tubule that is present in the near the Bowman's capsule. Proximal part means anterior part. Okay. The proximal convoluted tubule is lined by a single layer of irregularly shaped, so they may be cuboidal or columnar in shape, epithelial cells that have microvilli. You can see here, these are the cells of proximal convoluted tubule. They have microvilli, a lot of microvilli, forming a prominent brush border. So these cells are also called as brush border cells. These cells exhibit the following structures: the apical canaliculi, vesicles and vacuoles, which function in protein absorption. So their function is to absorb the protein. Prominent interdigitations along their lateral borders, which interlock adjacent cells with one another. And also they possess numerous mitochondria compartmentalized in the basal region. You can see here all the mitochondria congregated at the base of the cell by extensive enfoldings of the basal plasma membrane, which supply energy for the active transport of sodium out of the tubule. So during the urine formation, the sodium is actively transported, and this function is monitored by the cells of the proximal convoluted tubules. The function of this proximal convoluted tubule is to drain the Bowman's capsule at the urinary pole of the renal capsule. It reabsorbs from the glomerular filtrate all the glucose, amino acids and small proteins, and at least 80% of the sodium chloride and the water. It exchanges hydrogen ions, H plus ions, in the interstitium for HCO3- in the filtrate. It also secretes organic acids such as creatinine and bases and certain foreign substances into the filtrate. The next structure that we are supposed to look into here is the loop of Henle. So you can see here a small loop-like structure at the base of the nephron. This is called loop of Henle. There are two limbs for this: an ascending limb, a descending limb, and an ascending limb. The descending limb of the Henle's loop is also known as the straight portion or pars recta of the proximal tubule. It is lined by a simple cuboidal epithelium that has a prominent brush border and is similar to the lining of the proximal convoluted tubule. So the structure of the cells of proximal convoluted tubule we have already studied, and the same cells are present in the descending limb of the loop of Henle also. Its function is to reabsorb, exchange, and secrete in a manner similar to that of the proximal convoluted tubule. All the functions of the proximal convoluted tubule will be performed by the descending limb of the loop of Henle too. The thin limb of the loop of Henle. The thin limb of the loop of Henle is composed of descending segments, or a loop and an ascending segment, all of which are lined by simple squamous epithelial cells possessing a few short microvilli. The nuclei of these cells bulge into the lumen. In juxtamedullary nephrons, the thin limb can be divided into three distinct portions on the basis of the shape of the epithelial cells, their organelle content, the depth of their tight junctions, and their permeability to the water. Ascending thick limb of the loop of Henle. The ascending thick limb of the loop of Henle is also known as the straight portion, passa recta, of the distal tubule. It is lined by the cuboidal epithelial cells that possess only a few microvilli, an apical nucleus, and mitochondria compartmentalized within the basal plasma membrane infoldings. It establishes a gradient of osmolarity in the medulla. The ascending thick limb returns to the renal corpuscle of origin, where it is in close association with the afferent and efferent glomerular arterioles. In this region, the wall of the tubule is modified, forming the macula densa, which is a part of juxtaglomerular apparatus. What is this JG apparatus or juxtaglomerular apparatus? This juxtaglomerular apparatus is located at the vascular pole of the renal corpuscle. So this part is called the juxtaglomerular apparatus or JG apparatus. The JG cells are modified smooth muscle cells that exhibit some of the characteristics of the protein secreting cells. You can see here these are the juxtaglomerular cells which are present at the outermost surface of the afferent arterioles, and they are modified smooth muscles having some characteristics of the secretory cells. The JG cells are located primarily in the wall of the afferent arteriole, but few may also be present in the efferent arteriole as well. The JG cells synthesize renin, a proteolytic enzyme, and store it in secretory granules. The main function of these cells is to store, secrete and store the renin, an enzyme which is proteolytic in nature. So along with these cells, there are some certain cells called macula densa cells. These macula densa cells are tall, narrow, closely packed epithelial cells of the distal tubules. So you can see here, this is the distal tubule, and the wall of the distal tubule is having some specialized cells called macula densa cells, which are very near to the juxtaglomerular cells. So this is called juxtapositioning. Juxtapositioning means positioning very near, one after the other, very close to each other. See, this is the distal tubule and this is the afferent arteriole. They are close very together, and hence the cells with specialized characters, specialized functions present at these positions are called juxtaglomerular cells, or the entire structure is called juxtaglomerular apparatus. These macular densa cells have elongated, closely packed nuclei that appear as a dense spots, the macula densa, by light microscopy. The macula densa cells may monitor the osmolarity of the blood and the volume of the fluid in the distal tubule and transmits this information to the JG cells via the gap junctions between the two cells. So though these two cells have different origins and different functions, they are interconnected by the gap junctions. So the osmolarity of the fluid present in the distal tubule and the water content in the distal tubule as well is sensed by these macular denso cells. They act as baroreceptors, and that signal will be transmitted to the JG cells, based on which the JG cells either secrete renin or stop secreting renin. The extraglomerular mesoglial cells. So we have seen there will be some mesoglial cells in between the capillaries of the Bowman's capsule. That's the glomerular. We have lot of extraglomerular mesoglial cells in between the capillaries. So there are some cells which are present outside the Bowman's capsule. You can see here some green colored cells. This present in between the afferent and efferent arterioles. These are called extraglomerular, present outside the glomerulus. That's why they are called extraglomerular mesoglial cells. These are also known as Polkisson or pole cushions or LASIK cells. This acts as a cushion in between the afferent and efferent arterioles, hence the name pole cushion. And they lie between the afferent and efferent glomerular arterioles. What is the function of these JG apparatus or juxtaglomerular apparatus? The function of the JG apparatus is to maintain blood pressure by the following mechanism. Now, what is the mechanism involved in maintenance of blood pressure? A decrease in the extracellular fluid volume, perhaps detected by the macula densa cells, and we have already studied this in the previous slide, that the function of the macula densa cell is to detect the changes in the osmolarity as well as the fluid concentration of the fluid present in the distal convoluted tubule. So that will be sensed by the macula densa cells, and they have the direct connection with the JG cells through tight junctions. So, gap junctions. So now, once the signal is trespassed, it stimulates the JG cells to release renin into the bloodstream. Now, renin acts on angiotensinogen. We have already seen that the function of renin is to cleave the protein, right? It acts as a proteolytic enzyme. Now, the renin acts on angiotensinogen in the plasma, converting it into angiotensin 1. So in capillaries of the lungs and elsewhere, the angiotensin 1 is converted by angiotensin converting enzyme, it's ACE, to angiotensin 2. Carefully observe here the fluid concentration, the decrease in the fluid volume in the distal convoluted tubule stimulates the JG cells to secrete renin, and renin in the plasma converts the angiotensinogen into angiotensin 1, and this angiotensin 1 is converted into angiotensin 2 by the action of an enzyme called angiotensin converting enzyme, or ACE, either in lungs or liver or any other organs. And this angiotensin 2 is a potent vasoconstrictor. That means it has the ability to constrict the blood vessels that stimulate the release of aldosterone in the adrenal cortex as well. So when vasoconstriction takes place, the adrenal cortex will be excited, so that the adrenal cortex will be stimulated to release aldosterone. And this aldosterone in turn stimulates the epithelial cells of the distal convoluted tubules. The aldosterone, which is secreted by the adrenal cortex, reaches the distal convoluted tubule now, and distal convoluted tubule, once it comes in contact with aldosterone, sodium and chlorine ions are removed from the distal convoluted tubule. So once sodium and chloride ions are removed, the water follows the ions, right? So once you remove the sodium and chloride ions, what happens to the water? The water follows the ions, thereby increasing the fluid volume in the extracellular compartment, which leads to an increased blood pressure. So this is how water is reabsorbed from the kidneys by the help of the angiotensin, and this entire mechanism is called renin-angiotensin system, or renin-angiotensin mechanism of control of blood pressure. There are several mechanisms by which our body maintains blood pressure, and one of the mechanism is through the JG apparatus. The distal convoluted tubule is continuous with macula densa. We have already seen that this is the distal convoluted tubule, okay, and is similar histologically ascending thick limb of loop of Henle. It is much shorter, has a wider lumen than the proximal convoluted tubule, and lacks the brush border cells. So there will be no brush border cells in the distal convoluted tubule. The function of this distal convoluted tubule is to reabsorb sodium from the filtrate and actively transport it into the renal interstitium. This process is stimulated by the aldosterone. It also transfers potassium and NH4 ions along with hydronium ions into the filtrate from the interstitium. The connecting tubule is a short segment. So this is the CNT, this is the connecting tubule, okay. This is the short segment between the distal convoluted tubule and the collecting tubule. So this is the collecting tubule, major collecting tubule, and this is the distal convoluted tubule. There is a small neck-like structure called connecting tubule, which connects the distal convoluted tubule to the collecting tubule, and is lined by two types of epithelial cells. The principal cells, you can see here the first the principal cells. The principal cells may have many infoldings of the basal plasma membrane. These cells remove sodium from the filtrate and secrete potassium ions to it. Okay, it's the function of the principal cell is to remove sodium and add potassium. The intercalated cell, the second type of cells present in the collecting tubule is the intercalated cells. These cells have many apical vesicles and mitochondria. These cells remove potassium from the filtrate and secrete H+ ion into it. The collecting tubules have a different embryological origin from that of the nephrons. They have segments in both cortex and medulla, and converge to form larger and larger tubules. So there will be cortical collecting tubules as well as the medullary collecting tubules. So if the collecting tubules are present at the cortical region of the kidney, then they are called cortical collecting tubules, and these are primarily located within the medullary rays, although a few are interspersed among the convoluted tubules in the cortex, the cortical labyrinth. They are lined by a simple epithelium containing two types of cuboidal cells. Again, the principal cells, which process a round central nucleus and a central cilium. It's a round central nucleus in the principal cell. And the intercalated cells, which are less numerous than principal cells and possess microplicae, or many infolds on the apical surface. You can see here there are many infoldings are there, and many numerous apical cytoplasmic vesicles. So a lot of cytoplasmic vesicles present in these intercalated cells. Next we shall study about the medullary collecting tubules. In the outer medulla, the medullary collecting tubules are similar in structure to the cortical collecting tubules and contain both principal and intercalated cells in their lining of the epithelium. So same principal and intercalated cells that were present in the cortical collecting tubules are also present here. The same structure remains. Okay. In the inner medulla, the collecting tubules are lined only by the principal cells, but only the difference is in the medulla, inner medulla, the collecting tubules are completely lined only by the principal cells. No other cells will be present here. The papillary collecting tubules are duct of Bellini. The papillary collecting tubules are large collecting tubules. Okay, you can see here these are large collecting tubules with 200 to 300 micrometers in diameter, formed out of converging smaller tubules. So when the smaller tubules converge, the larger tubules are formed. They are lined by simple epithelium composed of columnar cells that have a single central cilium. They empty at the area cribrosa, a region at the apex of each renal pyramid that has 10 to 25 openings through which the urine exits into the minor calyx. So the function of the papillary collecting tubules are the ducts of Bellini. This region is called duct of Bellini, okay, or papillary collecting tubules. So once they collect the urine from all these smaller tubules, they empty this into area cribosa of the kidney, and the area cribosa is nothing but the apex of each renal pyramid. The tip of each renal pyramid is called area cribosa, where these papillary collecting tubules drain the urine, and each renal pyramid has about 10 to 25 such openings through which the urine exits into the minor calyx. And once they reach the minor calyx, from there they reach the ureter and finally reach the urinary bladder. So this is how urine is formed inside the kidney. So for any further information about the structure and anatomy and histology of the kidney, I would recommend you to refer these textbooks. Thank you.